Okay, I'm going to go through the exam pack on factors affecting photosynthesis, and as always, just a quick review of the objectives because they can guide you a little bit. So a little reminder, this is about factors affecting photosynthesis, that's CO2, that's kind of a bit obvious, and light intensity and temperature, but also the effects of water stress. And you should be able to carry out practical investigations into this as well, which is, we have some typical examples, and the examiner always gives you some stranger ones. Watch out for those too. Right, the first one question, as you may have seen, is either really obvious and you got it, or you've fallen down the obvious red herring hole that they will after you to get. So it talks about a pond plant with goldfish and more pond weed at the surface of water on a sunny day, but less so on a cloudy day. Give a reason for this difference. Well, some people will give the answer: the plant moves to the top to get more light on a sunny day. Clearly, plants can't move, or they have no control over that, so that will be the obvious wrong answer. Actually, it's thinking a little bit about photosynthesis and what's produced. We produce oxygen, and those oxygen bubbles can be trapped inside the leaf, because you remember that spongy mesophyll layer when we look at the side of a leaf, and then there's the palisade layer. Well, that does allow leaves to become more buoyant, and so the plant would rise up to the surface. The next question is a graph you may want to have seen, particularly useful to farmers because it helps them kind of gauge how fast a plant's growing. A little reminder that plants respire because they also carry out many processes like active transport. And it's just about the rate of production of carbohydrates of photosynthesis and how much is used in respiration. As always, before you approach the question, have a little think about what it means. So more carbohydrate is either in this case being used in respiration or being made in photosynthesis. I think I've got time of day. And the plant seems to start photosynthesizing around sort of 7 and finish about 4, and the plant is respiring all day long, but it does change. Right, first question says explain the shape of the curve. So again, lots of people might start describing it, and when we're at photosynthesis, there will be no marks for describing, especially when it's only 2 marks. They want you to give a reason why it goes up and potentially why it goes down. But I think in this case, that is just the opposite answer. So why does it go up? Well, clearly there is an increase in light intensity as the day and the sun rises. And that's mark worthy because I don't mention the light, you've got to infer that. And if it's more light intensity, well, it says about rate of photosynthesis, so there must be more of the light dependent reaction taking place. It says explain the shape of the curve for the rate of respiration. Again, curve, and you may have seen flatter versions of this. Just look at this closely. If we look, it's actually skewed. It is much on this side, a little bit higher than it is on this side. So what are the possible explanations for plants? Increasing respiration as we go throughout the day, what also changes as we go throughout the day, and what might also be slightly greater in the afternoon. And hopefully, if you think back to all those things that affect photosynthesis, and this is respiration, but clearly there was temperature. Why does temperature matter in respiration? Because it's an enzyme driven process, like all chemical reactions. So I'll be looking at temperature and enzymes for that answer. And finally, what's happening at point L? Well, L, if you look very closely, the usage and production of carbohydrates is perfectly matched, and that point is known as the compensation point. What that means is a point at which photosynthesis and respiration perfectly match. For a farmer, if you're below the compensation point, I mean, if your curve for photosynthesis was like this, you wouldn't be producing, your plants aren't getting any bigger, in fact they're probably getting smaller because they're using up carbohydrate they've stored, and this kind of does happen in the wintertime. But as the summer goes on, this bit here, the area under that curve there is the net production of sugars, and that's important if you're a farmer trying to grow stuff. Okay, so this is a topic I often talk about when I'm teaching this, and this is the idea of different types of plants. And it says they're growing different temperatures, which are suited to their metabolism. And they're adapted for growth in cool climates and grow well in warm climates. Plants also vary in their photosynthetic metabolism, so that's kind of the rate of reactions. Some plants produce a free carbon compound, that's the ones you should know about, it's your typical European crops, and they're called C3 plants. A group of other plants have adapted their pathways a little bit, so they are less sensitive to temperature, and they're called C4 plants. And it gives some examples here, things like barley and so on, and it can give some other examples of some C4 plants, again, more tropical ones. Figure 18 shows the assimilation, a bit like the Borg in Star Trek, they assimilate people or put things together. So this is about putting together carbon dioxide into larger carbohydrates. Right, again, get your head in the graph before you launch into the question. I know you want to rush off sometimes, but this is where the mistakes happen. So, assimilation of carbon dioxide per kilogram per hectare per hour. This gives you an idea of the rate. And clearly, it's going up as the temperature goes up. But also, not as it go up, it then decreases when we get to a certain point. It sometimes helps us label things. So these are my C4, and these are my C3 plants. So, ask about general relationships. You can do that. Make sure we talk about the up and the down. Calculate the values for the mean assimilation of carbon dioxide by C3 plants and C4 plants at 20 degrees. So again, you can calculate a mean. Just make sure it says include units in your answer. So that's where some people will drop marks a little bit. So once you know the mean assimilations, just be very careful about that. Suggest a conclusion that if we draw from the means, again, you can work on that. It's a 1 marker, so it's quite short. Okay. The next one says, with reference to figure 18.2, suggest which curve corresponds to each of the following crops. Sugarcane that grows in warm climates and barley which grows in cool climates. Well, the C4 ones were the ones growing in the warm climate, C4 ones were growing in the warm climates and the C3 in the cold climates. But I always like to just second guess myself and think, am I launching in a bit too confident here that I know the answer? What other info was given? Well, they want to know which curve, and I can only said perhaps one about crops as well. I don't know. Haven't looked. So which is the coolest one and which is the warmest one? We've asked us the extremes. So I'm going to go for crop. So dash dot dash. That's one. Crop 1 for C3. And crop 2 for C4. So I think that's kind of important, just not to launch in there, trying to see what else the examiner might be expecting. The worst is it just says ignore crop and we don't need to worry. But I just, kind of known examiners, I'd expect it to be there. Next one says temperature is very important in determining plants' ability to photosynthesize effectively. Temperature stress has become a great concern because of climate change. Higher temperature stress is defined as a rise in temperature sufficient to cause irreversible damage to plant growth and development. Some of the stress effects of temperature have been recorded in various plants and are outlined below. So moderate HT stress and severe, and we've got a list of different bits here that have changed. Again, things you probably haven't heard of. As the question says, assess. Now that's a new kind of sort of starter word. It's kind of like discuss or give a few reasons the impact of moderate HT stress on process of photosynthesis. It's 3 marks, so fairly logically, it's going to be 1 for each of these 3. So rubisco, heat deactivation, or no rubisco, no carbon dioxide fixation. That's about putting together your CO2s into a sugar. No change in chlorophyll fluorescence in photosystem 2. So fluorescence is when you've got an atom with an electron in it, and light hits that electron and it jumps up and it goes into a higher level shell. And then it will fall back down and give out that energy. In photosynthesis, that energy is not given out as different colour light, it's given out and passed on to chlorophyll A ultimately, and then it can be used to drive proton pumps and so on. So that's what fluorescence is. It says there's no change in the chlorophyll fluorescence, which seems a bit odd because this is all about damage. So I'm kind of guessing that actually it can't do this process. I'll be honest with you, that's a really badly worded statement compared to what the mark scheme is. So it's about the fact if it can't carry out the light dependent stage, what can it produce? So I'm going to say no NADP can be produced and ATP in light dependent reaction. And lastly, reduction in stomata. Well, what happens then? They let CO2 in, so there's less CO2 for carbon dioxide fixation. Again, write it out in full. You have 2 ways in which the ultrastructure of a chloroplast can be altered by high temperatures. So ultrastructure, we come across that in cells, and the chloroplast is a cell, even though it lives inside a plant cell, can be altered by high temperatures. Again, for each one, so we've got a suggestion and an explanation. And again, this is, so there are only so many things in terms of ultrastructure, so many things that you know of inside a chloroplast, and obviously it's made up of membranes. So cell membranes are going to be affected. So that's going to be our sort of theme. Is it anything else that might be affected within this ultrastructure? Well, ultrastructure, strictly speaking, is organelles. I do happen to know in this one that they talk about pigments, and strictly speaking, I would argue that's not part of the ultrastructure. They do go into it. But what if the cell membranes were affected? What's the effect of cell membranes being disrupted? And this can be a bit more detail. Well, on the cell membranes, we've got our photosynthetic pigments. So we reduce their ability to capture light. We know that the cell membranes within the chloroplast also maintain hydrogen ion gradients. So have less ability to produce ATP. And again, enzymes are also involved in the light dependent stage and compartmentalized. So this really is a question about the roles of cell membranes and the effects of temperature, which comes up at AS, but they've sneaked it in here, and this is a really typical applied question. So looking for things like that, and hopefully recognise it is about cell membranes. I always, when I'm doing these, I always try and look for different answers, and so I did spend a lot of time thinking this one over because my answers were all sort of cell membranes. I'm just sort of concerned the examiner might be looking at other organelles within the chloroplast, but it was fine, it was just cell membranes. And then finally, palisade mesophyll cells, for example, a specialised plant cell, and they're the main site of photosynthesis in plants. Team of scientists investigates the effects of shining light on the upper and lower surfaces on the rate of photosynthesis. And there's the results. So for each one, I've got the upper one on top and the lower ones. The upper side is photosynthesizing more. Any particular light intensity. What conclusions can you draw in the data from figure about the effects of light and the internal structure of a leaf? So it's quite hard. So really what we're asking is, why is it photosynthesizing more effectively? Now, you might, in your mind, be thinking about stomata on the lower side, but that's not really relevant to light intensity and not particularly relevant here. So what we need to do is sketch out that internal structure that we know. Spongy mesophyll, palisade mesophyll, and then it starts to lead you perhaps to discussing the palisade cells, and that's where you're linking to that by sketching out that diagram. Right, well done. Okay, please just reflect on some of the strategies I've used that might help you not make the silly mistakes. No one's saying I've got it perfect, but it's those strategies I'm trying to encourage you to get. Well done.